Hi, my name is Jennifer Shelton, and I'm going to be talking today about uncovering the phylogeny of plastids, specifically chloroplast, using bioinformatics. Before I begin, I'm going to do a quick run-through of endosymbiosis. Primary endosymbiosis is believed to occur when a free-living cyanobacteria is engulfed by a non-photosynthetic eukaryotic cell. One thing to remember here is that free-living cyanobacteria originally had its own genome, and a lot of that is maintained after the endosymbiotic event. Secondary endosymbiosis is when a free-living eukaryote that already has chloroplasts in it, or plastids in it, is engulfed by a second eukaryote. In the lineages resulting from that, you end up with a scenario where you have the engulfing host's nucleus, as well as genomes in the mitochondria, and a genome in the plastids. So we can use those genomes to create a gene phylogeny similar to the ones you see in your text. And that's exactly what we're going to be talking about today. Specifically, we're going to be talking about a Martin et al. 1998 plastid phylogeny. And we're going to be using the National Center for Biotechnology and Information, NCBI's website, to test this phylogeny. So I'm not going to get into too much detail of what Martin did yet. Before I do that, I want to talk about the basics of interpreting a tree. So at the base of any tree is the root. The root is generally a primitive distant relative of all of the leaves on the tree. The leaf nodes are at the top of the tree, and those are generally the taxa that we're specifically interested in. So say we're making a phylogeny of higher mammals, we might use a marsupial as a root because it's more closely related to a primitive distant relative, and it shares common ancestors to everyone on the tree. Internal to those leaf nodes, we have internodes. These internodes represent the last common ancestor between any two leaf nodes. So they would represent the last common ancestor between, say for example, two higher primates, which might be something more like what we see today as a lemur. Taking a look back at Martin et al.'s tree, our tree is rooted with cyanobacteria, which makes sense because we think that cyanobacteria, or primitive cyanobacteria, was originally phagocytized during a primary endosymbiotic event. For the leaves of our tree, we have some taxa that we might be more and less familiar with. So we have glaucophytes, which have plastids and are considered to be basal in this lineage. We have red algae, which we're pretty familiar with. And then over here, we have liverworts, which is a lower land plant, gymnosperms, dicots, and monocots, so all the way up to higher land plants. We've got a branch here off of about where red algae would be, up to euglena. Sorry, a branch here off of about where green algae would be, up to euglena, and a branch here off of the red algae branch, up to diatoms. These inner symbols here represent endosymbiotic events. So the primary endosymbiotic event occurred here when the ancestral eukaryote phagocytized a free-living cyanobacteria, and all of these lineages are believed to derive from that in terms of their chloroplast genome. And we've got secondary endosymbiosis when a red algae was phagocytized by a precursor to diatoms. We've also got secondary endosymbiosis of a green algae by a precursor to euglena. So euglena's plastids are believed to be derived from green algae, and diatoms' plastids are believed to be derived from red algae. So how did the authors of that paper make that tree? There's sort of four basic steps to making a tree. The first step is to find similar sequences. So we're talking about a gene tree here, or a protein tree. So these are going to be sequences of either nucleic acids or amino acids. And we're looking for similar sequences in our large databases to begin with because we're looking for potentially related genes and proteins. Once we find similar sequences, we do something called an alignment, where we line up the similar sequences. And then for our third step, we consider the dissimilarities between these similar sequences. Sounds a little bit strange, but what we're looking for here is who is most closely related to who. So in this example, for example, we've got 6 letters. We start with piggies. We've got a sequence that only varies from piggies by 1 amino acid. And then we've got a sequence that varies from piggies by 2 amino acids. So altogether, the first and the third sequence are the most dissimilar from each other, and the second sequence is equally similar to both the first and the third. So our fourth step, after we've tallied our dissimilarities or number of mutations, is to take those relationships and make a tree that explains the relationships between these sequences well. So if we made a tree out of this, we would want the first and the third sequences to be farthest apart from each other. For our first step today, we're going to use BLAST, which is Basic Local Alignment Search Tool. That's a freely available NCBI program that's designed to search large databases for biologically relevant sequence similarity. So we're going to use that to find our potential related genes. And then we're going to use the distance tree tool on NCBI to make our own tree. The gene that we're going to be generating a phylogeny from is PSBE. That's part of photosystem 2. It's considered critical to photosystem 2, so we should see it in definitely photosynthetic organisms. In the Martin et al. 1998 paper, they used PSBE as one of 45 genes that they used to make their tree. So we're going to be doing a miniature version of their experiment here today. And before we begin, I just want to take a moment and think about what our hypothesis might be. So we're looking for how similar the tree that we create with our gene is to the tree that Martin et al. Created. But what kind of dissimilarities might we be trying to avoid? Specifically, I'd like to see lineages like what we see in this tree. So yeah, I'd like to see cyanobacteria showing up, red algae, green algae, land plants, glaucophytes. I'd like to see all of the same leaf nodes and roots, but I wouldn't like to see mammals, reptiles, insects, because we think that these genes occur in eukaryotes as a result of endosymbiosis of cyanobacteria. And mammals, reptiles, insects, for example, don't have chloroplasts, so we wouldn't expect to see them showing up there. So with that, I'm going to shut this down a little bit.